Hello, my name is Dhruvesh, and I'm the president of the bioengineering team here at Tuscarora. We're focusing on oyster recovery in the Chesapeake Bay, and we're basically trying to create a matrix which can trap a parasite which has been harming the oysters for a long time.
Oysters are very important to the Chesapeake Bay. They filter the water and they filter out phytoplankton that could harm other organisms. The oyster populations have been on major decline recently, and this is mainly due to over-harvesting, pollution, and disease. We are focusing on the disease aspect.
There are many diseases that are affecting oysters. However, the one that we chose to focus on is known as dermo. Dermo is caused by Perkinsus marinus, which is caused when Perkinsus marinus infects oysters by attaching to the galectin receptors that are found on the cell membrane. It enters the oysters and replicates inside the cells. After it replicates in the cell lyses and it splits open. By the time that it finishes with replication, it causes the inside of the oysters to deteriorate. The spores tear open the host, and this causes the death of the oyster. This happens over a long period of time, and after the host dies, the spores are released into the water, which allows the cycle to begin once again. How can we help? We plan to capture this spore before they can reach the oyster. We are trying to manufacture a synthetic seaweed, which will have this galectin receptor on its surface. With this, we will be able to capture these spores by making sure that this spore attaches to the protein receptor on the seaweed instead of attaching to the protein receptor on the cell of the oyster.
So last year we produced our first batch of cellulose using xylinus that we received from Imperial College. Xylinus is the bacteria that produces the cellulose, and you can see the cellulose sheet performed in this test tube. And we also made a methods paper for producing the xylinus. It's called a method for producing cellulose using the bacteria Gluconacetobacter xylinus.
This is a model that shows our framework for creating the seaweed. So we have the cellulose sheet. To it we will connect a fusion protein, which will consist of a cellulose binding domain protein and a galectin protein. And the galectin protein is a receptor for the P. marinus disease, and we hope that once we have created it, it will attract the P. marinus and protect the oysters.
Okay, so this process is done mostly in two parts, the galectin and then the cellulose binding domain. University of Maryland has already worked with this galectin and identified the galectin protein on the oysters. So we'll be using that sequence from the Adjunct Part Registry. And Imperial College has already been working with the cellulose binding domain, and they were kind enough to help us out and send us the xylinus, and we were able to use the cellulose binding domain because of them. We're creating an operon that has the galectin and the cellulose binding domain eventually, and which also uses the BFP protein, Blue Fluorescent Protein, that will be used as an indicator so we can identify if the transformation actually works. So we're fusing them together to make one full fusion protein. By doing this, we created our own sequence and sent it off to be produced. It started with a promoter, a ribosome binding sequence, and then started off with the cellulose binding domain and the GFP. There was a base pair restriction, so we couldn't also include the galectin in our original one that we're producing. So right now we're just focusing on attaching the cellulose binding domain and also the blue fluorescent protein together in one fusion protein. Eventually we want to create one functional operon that will include both parts.
When we get the parts to make the fusion protein by the cellulose, we will conduct an experiment where there are two there's an experimental group and a control group. They're both the same, except one has the cellulose with the fusion protein, which is the experimental group, and one has the cellulose without the fusion protein, which is the control group. We will leave them overnight, and the next day we'll spec them and see how much P. marinus there is. The experimental group should have less P. marinus.
We would like to give a special thanks to the Imperial College of London for sending us this xylinus, the University of Maryland for isolating the galectin gene, our team mentors, parents, and Ars Biotechnica for helping us do this whole project.